Hello student. Now we are going to start blood physiology. The first thing that we are going to understand here is hematopoiesis, process of generation of mature blood cells. So all of you know that this is a complex process, but it's an orderly and multi-layered process by which matured blood cells are generated from hematopoietic stem cells. So this process we are going to understand first. First thing is when the hematopoiesis start for the first time, or what are the organ where hematopoiesis is occurring. In human, it is said that hematopoiesis start in and around day 19 of gestation. In and around 19 days of gestation, right? So that is the first time when hematopoiesis is seen in human, right? And the organ from where the hematopoiesis is occurring is nothing but, all of you know, that yolk sac, right? Along with yolk sac, up to this third week of gestation, hematopoiesis also occur at the level of mesodermal structure, like for example, aorta, gonads, and mesonephros. Then, this hematopoietic activity shift from this yolk sac and mesodermal region to the level of liver, spleen, and lymph node. So, in and around 3 to 4 months of gestation, the major hematopoiesis occurs from the level of liver as well as your spleen and lymph node. Then, the hematopoiesis will shift from this organ to the bone marrow, right, where the adult hematopoiesis also occur. And this is known as the major switchover. The major switchover of hematopoiesis occurs from this liver to bone marrow in and around 20 week of gestation. So this 20 week will be an MCQ. You have to remember that the major switchover of hematopoiesis from liver, spleen to bone marrow occurs in and around 20 week of gestation and at the level of birth. So obviously this conversion, this switchover has occurred in 20 week of gestation. So at point of birth, the major organ for hematopoiesis is nothing but your bone marrow. Now, bone marrow will continue your hematopoiesis throughout your life. But you know, initially, the long bone will also take part in hematopoiesis. But during the process of aging, when the person will reach at the level of puberty, the hematopoietic activity of the long bone will be decreased because the long bone marrow will be converted into fatty deposit, except the terminal portion of the long bone. The most of the part of the long bone marrow will be converted into fatty tissues, because of which the long bone will lost its hematopoietic activity. After puberty, hematopoiesis will only continue in membranous bone in our body, that is vertebra, sternum, ribs, and iliac bone, right? So this information has beautifully depicted in this diagram, which I have taken from Winthrop's textbook. So in x-axis, you look at this is the point of birth. So this line is the point of birth. So as you are finding out here, on your left hand side, this is the fetal month before birth, and on your right hand side, this is the after birth, the age of the person in years. So before birth, okay, initial stage, the hematopoiesis is occurring only at the level of yolk sac in and around 19 days, right? And this yolk sac hematopoietic capacity is gradually decreasing and it is vanishing in and around this 2 months of gestation, right? But by the time when the yolk sac activity is decreasing, the activity, hematopoietic activity at the level of liver is rising to a very high level. Similarly, the hematopoietic activity of the spleen is also rising and the lymph node hematopoietic activity will also rise. But you look at this liver, spleen, they are maintaining their hematopoiesis up to this point, means up to this 4 to 5 months maximally. But after that, the hematopoietic activity of both of these liver as well as spleen is decreasing. And before birth, the hematopoietic activity of liver is completely finished, as well as the spleen has stopped its hematopoietic activity long ago, right? So, at the point of birth, the main hematopoietic activity is continued at the level of bone marrow only. So that's what we're finding out here. And bone marrow has started its activity in and around 20 weeks of gestation, the major switchover point. then the activity of the bone marrow is increasing, and at the point of birth, it is the major organ of hematopoiesis. Which bone? Both long bone and membranous bone. But when the age of the person is increasing and the person is reaching in and around puberty and little after pubertal age, the hematopoietic activity of the bone marrow, particularly of the long bone, like for tibia, like for example femur, their hematopoietic activity is reduced because of the fatty deposition inside this bone marrow. But throughout the life, after this 20 years of age or after this puberty, the hematopoietic activity is only continued in membranous bone, like for example this vertebra, sternum, ribs, right? So this way, this diagram is beautifully depicting the timeline of hematopoiesis. Now after this, we are going to understand that in case of adult, we came to know that bone marrow is the main site of hematopoiesis. Where inside the bone marrow the hematopoietic activity is occurring, right? So on your left hand side, this is a diagram where you are finding out this is the cross section of a bone, where finding out your periosteal artery you are getting and the central artery you are getting, and this central vein you are also finding out. Now there is a venous sinus. Okay, there is a venous sinus here. Okay, so this sinus, in between this sinus region, the hematopoietic stem cells are located. So the stem cells are located inside, in between this, okay, hematopoietic sinus, not inside the vessels, but outside the vessel, but close to this venous sinuses. That's why it is said that hematopoietic stem cell, they mainly resides in the perivascular space, particularly in the region of the trabecular bone. So perivascular space. If this is the blood vessels of this sinus, just outside this blood vessel, your hematopoietic stem cell will reside. Okay. If body demands, or suppose bone marrow is suffering from certain disease condition, like for example fibrosis or cancerous cell is growing inside the bone, then this hematopoietic stem cell will enter inside this blood space, enter inside this blood, and it will circulate in your body, and it will be deposited in extramedullary site, like for example your liver and spleen, and that is known as the extramedullary hematopoiesis, right? So main location of this hematopoietic stem cell in adult is the perivascular space. But apart from this perivascular space, certain hematopoietic stem cell also remain close to this endosteal region of the bone, close to this endosteal region of the bone. Okay, so that is known as the endosteal region hematopoiesis, right? So one is hematopoietic stem cell close to the vessels, that is a vascular region, and one is hematopoietic stem cell close to the endosteal surface, that is endosteal region hematopoietic stem cell. The area where the hematopoietic stem cell is located, that area is known as hematopoietic niche. The word that we used is niche. Niche is nothing but it's a community. In that community, multiple cells resides, not only hematopoietic stem cell, although he is one of the main component, okay, but apart from this hematopoietic stem cell, other cells are also there, like macrophage, like fat cells, okay, like your osteoblast cell, osteoclast cell, everybody is there. So all these cells, they are making a community, okay. Then this community, they are living happily and they are helping one another. Means they will communicate signals between them. They will secrete, one will secrete certain growth factor that will affect other, that will secrete certain factor that will affect other cells. So this way, they are maintaining a community, and this community is helpful for proliferation as well as differentiation of the hematopoietic stem cells, and this community is known as hematopoietic niche, right? Or this is also known as hematopoietic inductive microenvironment, hematopoietic inductive microenvironment, right? So hematopoietic niche is located in two areas. One is the perivascular area, like here, and one is the endosteal niche. perivascular niche and endosteal niche. This endosteal niche is mainly important during the stem cell transplantation. During stem cell transplantation, you know that stem cell transplantation commonly which is known as the bone marrow transplantation. So whenever stem cell is transplanted, first some preconditioning regimen is given in the form of chemotherapy or radiotherapy. So whenever you are giving chemotherapy or radiotherapy, they generally damages this vascular niche. Okay, this vessel, the blood vessel, the endothelium cell, they will be damaged. So now after damaging of this vessel space or endothelium space, when you transfuse this stem cell into the donor, the stem cell will not get its niche at the level of this vascular area because that is already damaged by chemotherapy. So just after stem cell transfusion, just after bone marrow transfusion, hematopoietic stem cells, they reside particularly close to these bone cells, close to the osteoblast, close to the osteoclastic cell, and that is known as the endoblastic niche. So you can say that after stem cell transplantation, initially the hematopoietic stem cell which is transplanted, they reside close to which niche? Endosteal niche or bone niche of hematopoietic cell, right? So now we have to understand that in the community of hematopoietic niche, what are the different cells? I told you that there will be multiple cells, like for example, mesenchymal cell and endothelial cell. Vessels are there, blood vessel, it is close to vessel. So obviously endothelium cell will be there and mesenchymal cell will be there. This endothelial cell and mesenchymal cell, both of them secrete one important factor, and that is KIT ligand. Please remember this name, KIT ligand. Not only that, it also secretes CXCL12. These are the two important factors for the proper development of hematopoietic stem cell in the community. There are multiple factors, but I'm telling you, all of these factors has not given in textbook, particularly if you look Harrison's. But these two factors are specially mentioned even in Harrison's internal medicine also, so they can be asked in your MCQ. So apart from this mesenchymal and endothelial cell, there are macrophages also, megakaryocyte also, osteoblast and osteoclast are also there, and they secrete various transcription factors like BMI1, microRNA processing enzyme like Dicer. All of these factors are also important for hematopoietic stem cell development. So in that community or in that hematopoietic niche, multiple factors are there which are secreted by various cells in the community, and they help in hematopoietic stem cell development. So again and again, I am saying hematopoietic stem cell, hematopoietic stem cell. So what is the meaning of stem cell to you? If you say that this cell is a stem cell, it has two important properties. One is self-renewal and other one is the differentiation. Self-renewal means stem cell has to be able to generate similar kind of cell without any differentiation to maintain its population intact, right? So suppose this is one stem cell, it is giving rise to another cell. If this cell contains the same property like that of the original cell, like that of the mother cell, then it will remain in the storage pool. It will remain in the pool of the hematopoietic stem cell only, and this is nothing but self-renewal. But suppose whenever during cell division, it is giving a cell which is completely different from the mother cells. Okay. So this cell which is completely different from the mother cell, it will follow a different pathway and it will not come back into the community again. So this is known as the differentiation. This is known as the differentiation. And because of more and more differentiation, ultimately your mature cell is going to form. So there is one which is self-renewal, because of which it is maintaining the population intact. Otherwise, there will be shortage of this hematopoietic stem cell, right? So in one way, you have to maintain your original population intact, and in other way, you have to also give rise to the mature cell by the process of differentiation. These two properties are essential for calling a cell as a stem cell. Now, it has been seen that stem cell, they divide in three major types. They can divide in three major ways, right? Look at. So this is hematopoietic stem cell. It can divide in one way where it will give rise to two stem cell. Exactly same to same cell, which is like that of the parent cell or mother cell. So this is the mother cell. Okay. So in one way division, it is giving rise to two cells, but both of the cell contain the same property like that of the mother cells. So this is symmetric division, but both of these cell will join the hematopoietic stem cell only. So this is the self-renewal process. In other way, in the second type of division, it can give rise to two cell. Both of these cells are different from the mother cell and they will follow a different path. This is known as the differentiation. Differentiation. And in third type, hematopoietic stem cell will divide in such a way that one of them will remain the same character like that of the mother cell and one of them will not maintain the mother cell character. So the cell which is maintaining the mother cell character, it will join the hematopoietic stem cell pool. So this is again your self-renewal, like here also self-renewal, but the other cell will follow a different path and this is nothing but the differentiation of cell. So always there is a balance between these three pathways. Always there is a balance between these three pathways in the body. If your mature cell demand is more, then this differentiation will increase, right? But whenever your hematopoietic stem cell reserve is going to be a little decreased, it will divide more in this fashion. So this way, hematopoietic stem cell is continuously dividing. Three types of division is possible. So first two types of division, this is known as symmetric type of division, first two types. And the third type of division, this is known as asymmetric type of divisions, right? So this has been added in recent edition of Harrison. Okay, although it was there in the research article, but Harrison has added this symmetric division of hematopoietic stem cell here. And in third type, this is asymmetric division of hematopoietic stem cell. Now, stem cell are of different types. One of them is known as totipotent stem cell. Here, one of them is pluripotent stem cell and another one is the multipotent stem cell. Although there are others also, precursor stem cell, right? Progenitor stem cell, I'll come to that. But before, what is this totipotent stem cell? Totipotent, T-O-T-I, totipotent. Totipotent stem cell means this cell is capable of producing the whole organism, including the placenta. What I'm saying, make it clear. So you know that after the formation of the zygote, there will be division, cell division, 2 cells, 4 cells, then 8 cells. If you take any cell up to this 8 cell stage, so suppose this is the 8 cell stage embryo. If you take any one of these 8 cells and if you give a complete supporting medium for the proper growth of this cell, you are giving a supporting medium, then this single cell can give rise to whole organism, means all cells of the organism, as well as it can also give rise to placenta. So it will give rise to placenta also. It will also give rise to the whole organism, means all cell of the organism. Suppose human, the whole human as well as the placenta can be formed from a single cell if you collect that single cell up to 8 cell stage of embryo. So up to the 8 cell stage of the embryo, individual cells are known as a totipotent stem cell. They are capable of generating whole organism, including placenta also. Very, very powerful cell, totipotent, very, very powerful cell, right? Now, we know that this 8 cell stage will further grow, and then there will be formation of a cavity inside this. So this is known as the blastocyst cavity, right? And then you are finding out here one cell mass. This is known as the inner cell mass, and outer to this, obviously this is known as the outer cell mass. Okay. Now this outer cell mass is going to form placenta, but if you collect any cell from this inner cell mass, they are also capable of generating whole organism, but they cannot generate placenta because the placenta is formed by this outer cell mass. So if I collect any of this cell from this inner cell mass stage, this inner cell mass has the capability to produce the whole organism, every cell of the organism, but it cannot form the placenta. So the power of this inner cell mass is a little lesser than that of the totipotent cell. So this kind of cell, which is collected from this inner cell mass, it is known as pluripotent stem cell. Means pluripotent stem cell can give rise to all cells of a whole human, whole organism, but not the placenta, right? Now this pluripotent stem cell will move further, will differentiate a little bit, and after differentiation, it will form a multipotent stem cell. Multipotent stem cells are a kind of committed cell. Committed. Okay. So previously pluripotent was not committed. It can generate ectodermal tissue, endodermal tissue, mesodermal tissue, everything. But now multipotent cell, it's a little committed one. Means it will say either I will form ectodermal component, or I will form mesodermal component, or I will form the endodermal component, but not all. I'm not that much open. I'm a little reserved. I'm a little dedicated, right? So this kind of committed cell, which can form only one lineage of cell, either ectodermal component or mesodermal component or endodermal component, then this kind of stem cells are known as multipotent stem cell. You know that ectodermal component means there are multiple ectodermal tissues in our body. All this ectodermal tissue can be formed from this multipotent stem cell, but this ectodermal multipotent stem cell cannot form the mesodermal component, and vice versa. This way, the multipotent stem cells are a little dedicated cell towards single type of cell. So our hematopoietic stem cell, they are pluripotent stem cell only, but from pluripotent, first it will form a multipotent hematopoietic stem cell, and then it will give rise to the other mature cell in the body. So hematopoietic stem cell, they are pluripotent. From pluripotent, it will be first converted into multipotent, dedicated, and then it will form the other blood cells, right? So let's see how this hematopoietic stem cell development occur, right? So first we told that totipotent, pluripotent, then multipotent. After multipotent, you also have to remember another stage, that is the progenitor stem cell. Then it will be converted into precursor cell, and then finally it will form a mature cell, right? So totipotent, pluripotent, multipotent, then your progenitor, then precursor, then mature. Now when this stem cell, right, is moving from pluripotent, totipotent, progenitor, precursor, mature, look at this arrow. When the cell is moving in this direction, the cell is becoming more and more differentiated, right? Means it is losing the original character more and more. It is becoming maturing, right? So this green color thickness, this green color triangle which is given here, it indicates that more you move from this stem cell towards this mature cell, your differentiation is more and more. Differentiation is increasing, right? But whenever you are becoming more and more differentiate, what you are losing this character. What is this? This is the self-renewal ability. This is the self-renewal ability. That's why we can find out that the self-renewal ability is maximum at the level of the stem cells, and then it is decreasing toward this progenitor stem cell. Look at this arrow. Look at this arrow, or look at this triangle. Okay. So maximum self-renewal capability is the stem cell. When it is moving toward the precursor stem cell, the self-renewal property is decreasing, but differentiation is increasing. And look at the third one, proliferation activity, right? So proliferation activity is high from the stem cell towards this precursor stem cell. Then the proliferation activity is decreasing gradually towards the maturation, right? Mature cell means their proliferation capacity is very, very less. So what we came to know from this diagram, more the cell is maturing, it is becoming more and more differentiated. It is losing self-renewal ability. It is also losing the proliferative ability. Now there are two cells which are present in your blood, which are the mature cell, but they contain proliferation capacity as well as self-renewal capacity. That is the exception to the rule. And this is nothing but lymphocyte. Okay, memory B cell and memory T cell. All of you know they have the ability to self-renew themselves and they have the ability to proliferate whenever their memory is activated. That way, you have to remember that this triangle is given here. This indicates self-renewal triangle, and this triangle is given here. This is the triangle of proliferation. They have the ability for self-renewal as well as proliferation. So this is the different stages of cell through which the stem cells are moving from stem cell to mature cell. Now, if you look at the whole picture, that how this hematopoietic stem cell is giving rise to multiple mature cell in your blood, this is the whole picture. So first you look at here, hematopoietic stem cell. You have to remember these cells in your memory, right? So you're finding out here, this is the hematopoietic stem cell, which is pluripotent in nature. Then it is converting into multipotent progenitor stem cell, right? That multipotent progenitor stem cell will divide either in the direction of myeloid progenitor or in the direction of lymphoid progenitor. So that's why it is known as common myeloid progenitor or common lymphoid progenitor, right? So it is moving either in the common myeloid progenitor or common lymphoid progenitor. Now this common myeloid progenitor, it is forming two cell division, two cell line. One is known as granulocyte monocyte progenitor series and other one is megakaryocyte erythroid progenitor series of cell, right? So myeloid cell, they are giving rise to two lineage. One is granulocyte monocyte progenitor lineage and one is megakaryocyte erythroid progenitor lineage. Now, obvious thing is that granulocyte monocyte progenitor means it will be divided into granulocyte colony forming unit and monocyte macrophage colony forming unit. Colony forming units. If you keep this cell in culture media under proper nutrition, the cells forms a colony in the culture media. That's why it is known as a colony forming unit. It is forming a colony of cells, right? So granulocyte colony forming unit and monocyte colony forming unit ultimately giving rise to the mature monocyte that is coming from MCFU. And from this granulocyte colony forming unit, we all know what are the granulocyte in our blood, that is neutrophil, eosinophil and basophils. Now in this picture, I will tell you, try to remember the cytokines that I have written here. For the proper development of neutrophil series of granulocyte, you require GCSF. Okay. For proper development of eosinophil series, you require interleukin 15, and for the proper development of the basophil series of cell, you require interleukin 3. Right. On the other hand, if you look at this megakaryocyte erythroid progenitor cell, so very simple, it will give rise to another two types, that is erythroid colony forming unit and megakaryocyte colony forming unit. Ultimately, this is going to form your RBC and this is going to form your platelet. Right? So this is the development of myeloid progenitor cell. Now on the other side, look at how the lymphoid progenitor cell developed. Lymphoid progenitor cell, it will be divided into two lineage. One is B cell progenitor lineage right here, and one is T cell NK cell, that is a natural killer cell lineage. So one B and one is TNK. In both the cases, the interleukin which you require is interleukin 7 for the proper development of the cell series. Now, this B cell will develop further under the influence of interleukin 4 and it will form the mature B cell, and the T cell NK cell will be further divided into T cell series and NK cell series. For the T cell series proper development, you require interleukin 2, and for the proper development of the NK cell, you require interleukin 15. Ultimately, they will give rise to the mature T cell, it will give rise to the mature NK cell, and it will give rise to mature B cells. So this way, the hematopoietic stem cells are developing in a step-by-step fashion. This diagram which I have taken here, this is taken from Addison's Internal Medicine. Different book, there is a little variation from book to book. So that's why follow the standard textbook from where you have to remember this, right? Follow a standard textbook, and it is from addition, so you can rely on it, right? Now, after discussion of this hematopoiesis, now we are going to discuss various mature blood cells, right, in brief, because they are not very important for our MCQ purpose. So we'll quickly go through these mature cells which are present in our blood. So first one is the RBC. All of you know that RBC is a biconcave in shape, okay? And what is the diameter and what is the thickness of this RBC? All of you know. So this diameter is 7.2 micrometer. This is important to remember. The surface area of RBC is 120 to 180 micrometer square, and the volume of a single RBC is 90 femtoliter. And the typical shape of the RBC is biconcave shape. All of you know, because of this biconcave shape, what is the advantage? There is increased surface area, because of which the gases exchange, like oxygen, carbon dioxide gas, they will be easily exchanged. It will give more surface area. Second, because of this biconcave shape, there will be easy folding of the RBC. RBC can get folded itself on itself easily. And because of this folding, it will easily penetrate through the capillary without any rupture. So this is the advantage of the biconcave shape of the RBC. Not very important for your MCQ. Now the development of the RBC is more important, right? So erythropoiesis, development of the RBC. So whenever you are reading this development of the RBC, please keep in mind that already we have seen the development of the hematopoietic stem cell. Then that hematopoietic stem cell will form the common myeloid progenitor or common lymphoid progenitor. So common myeloid progenitor cell, and from that common myeloid progenitor, your erythroid megakaryocyte progenitor will form, and from there, the first cell in the RBC series. The first cell in the RBC series development is the pro-normoblast or pro-erythroblast. So the same cell is also known as pro-normoblast or pro-erythroblast. If you look at this cell, then this is a very, very big cell with bluish color cytoplasm and hugely enlarged nucleus. I think the size of the nucleus is very, very large, right? Now, if you go from this pro-normoblast to early normoblast, intermediate normoblast, late normoblast, then reticulocyte and mature RBC, what you are finding out that the cell size is decreasing gradually. Nucleus size is also decreasing, but cell is becoming more and more pinkish in appearance. So during this process of erythropoiesis, what is happening? There is decrease in size of the cell. There is decrease in size of the nucleus, but there is increase in hemoglobin content without the RBC, because of which it is taking more and more pinkish color, right? So the first cell is the pro-normoblast or pro-erythroblast. Then it is forming early normoblast, which is also known as basophilic normoblast. Basophilic normoblast. Intermediate normoblast is also known as polychromatophilic normoblast. Late normoblast is also known as orthochromatophilic normoblast. Okay, normo or erythro, whatever you say, right? So size is decreasing gradually. Now look at the other things. Look at the other thing. So pro-normoblast means the cytoplasm deeply basic in nature. Deeply basic in nature. Same for the early normoblast. The cytoplasm is basic in nature. Now, hemoglobin will appear in adequate amount in the intermediate normoblast stage. Adequate amount. Why I am saying this word adequate amount? Because if you look at hemoglobin, this is very, very important for your MCQ. If you look at hemoglobin, it is present even in the pro-normoblast cell also. It is present even in the early normoblast stage also. So if the question is hemoglobin start appearing in which stage, your answer is pro-normoblast or pro-erythroblast. But this amount of hemoglobin which is present here is so minute that the Giemsa stain or Romanesque group of stain will not be able to stain this hemoglobin. Hemoglobin takes pinkish type of color. So it will not be able to stain this hemoglobin. The amount is so less. So here the staining character of hemoglobin, that is the pinkish color, is not going to be seen. Adequate amount of hemoglobin for the first time is appearing here. What do you mean by adequate? Adequate means adequate which is stain with Giemsa or Romanesque group of stain. So because of the adequate hemoglobin, within this basophilic, within this bluish cytoplasm, some pinkish color will start appearing close to the nucleus. Getting my point? Right. Whenever this pinkish color, because of hemoglobin, is appearing inside this bluish cytoplasm, that is known as polychromasia, polychromatic cell. There will be different hues of color. Okay, pink color, in between deep blue color, pink color. So this polychromasia is seen in this RBC or RBC cell series, intermediate normoblast, due to appearance of huge amount of hemoglobin in this stage. That's why this is known as polychromatophilic normoblast or intermediate normoblast. Then the hemoglobin content will increase more and more towards more maturity. Okay, so whenever hemoglobin is becoming more and more, cell will take more and more amount of pink color, pinkish stain, right? That's why in the late normoblastic stage, pink color is so much that the cell will looks like reddish pinkish in color, and that is known as orthochromatophilic cell or late normoblastic cell, right? After this, what happens? Hemoglobin will still go on rising, but now the cell will lost its nucleus completely, and this cell is known as the reticulocyte. So please note down, this is the first cell where nucleus is completely lost. Even before this cell stage, nucleus is there, but that is small sphygnotic in size. In the reticulocyte cell stage, the nucleus is not there. Cell contain huge amount of hemoglobin, as well as reticulocyte also contain certain amount of RNA and certain amount of ribosome. If you stain this reticulocyte, okay, reticulocyte stain means if you stain this RNA and ribosome with a special type of stain that is known as supravital stain. You know the example of supravital stain, the new methylene blue and brilliant cressyl blue. New methylene blue and brilliant cressyl blue. These are the two supravital stain which are specially used to stain the RNA and ribosome which is present inside the reticulocyte cell. So if you stain this reticulocyte with this supravital stain, you are going to find out a net-like structure. This net-like structure is due to ribosome and RNA. Because of this net-like structure, the cell is known as reticulocyte. It contains reticulum net, right? And after this reticulocyte, the mature RBC will form with whole hemoglobin inside it. So it will take a full pinkish color, right? So this way, the development of RBC is going on. So we have to remember, more the development, more the maturity, the cell will lose its nucleus, the cell will lose its nucleus, the cell will decrease in its size, as well as there will be more and more hemoglobin, right? So this is the development of RBC. Now, erythropoiesis require one important factor, not factor, you can say important hormone, and that is nothing but your erythropoietin. So erythropoiesis require many factors, but one of the important factor is erythropoietin, EPO. Erythropoietin, you know where from it is produced? EPO, erythropoietin, produced from kidney, obviously kidney, but which cell of kidney? Okay, it's very confusing. Sometimes you mixed up with the JG cell. JG cell is for renin, it is not for erythropoietin. Remember that erythropoietin is produced from both kidney as well as little amount from liver. At the level of kidney, it is formed from the peritubular epithelium cells. Okay, so peritubular epithelium cells, right? So you know that there are tubules, and surrounding the tubules there are peritubular capillaries. Surrounding these peritubular capillaries there are epithelium cells. So that's why I'm saying here the peritubular capillary epithelium cell or peritubular epithelium cell. Peritubular capillaries epithelium cells, they are the major source of erythropoietin. Okay, so the epithelium cell which are present surrounding the capillary, peritubular capillary. And in case of liver, the peritubular venous hepatocytes are the source of 15 to 20% of total erythropoietin in our body. So the major source is from kidney. And the major stimulus for erythropoietin secretion, we also know that hypoxia is a major stimulus for this erythropoietin secretion, right? Whenever erythropoietin is secreted, we all know that it is going to stimulate the RBC development. RBC number will increase in your blood. So how erythropoietin increases RBC number, increases RBC proliferation? Do you think that it acts at the level of hematopoietic stem cell? Answer is no. Do you think it acts at the level of common myeloid progenitor? Answer is no. Do you think it acts at the level of megakaryocyte erythroid progenitor? Answer is no. There is no action of erythropoietin up to this level, neither hematopoietic stem cell or common myeloid progenitor or megakaryocyte erythroid progenitor. No. Erythropoietin starts its action from burst forming unit. So this BFU, we have already told what is colony forming unit. Burst forming unit is similar to that colony forming unit, but more immature type of colonies. So that is known as burst forming unit, BFU. Erythropoietin starts its action from BFU. It also stimulates this CFU. It also stimulates this early pro-erythroblast. It also stimulates this basophilic erythroblast, okay, polychromatophilic or erythroblast, but no action again at the level of this orthochromatophilics or reticulocyte or erythrocyte. So now I can understand that erythropoietin is only acting at the level of BFU, CFU, pro-erythroblast, basophilic erythroblast, and polychromatophilic erythroblast, not at the level of even ortho, reticulo, or mature cell also. But if I have to choose only one answer, that where is the major action of erythropoietin, then you have to look at the receptor. So erythropoietin acts on its receptor, which is present on all of this cell. Receptor is present on all of this cell. But if you look at the density of the receptor, maximum erythropoietin receptor is present at the level of CFU. That's why the main action of erythropoietin is at the level of CFU, colony forming unit, erythroblast. So this is the important MCQ that you have to remember, right? Very, very important, right? So this is all about the erythropoiesis or generation of the RBC. Now inside the RBC, we are again and again talking about the hemoglobin. So if you look at the structure of the hemoglobin, okay, this is very simple. All of you know that it is composed of 4 heme and globin, okay, globin chain. 2 alpha, 2 beta globin chain is present in adult hemoglobin. Remember, combination of this 2 alpha, 2 beta means 4 globin chain as a whole is known as polypeptide globin. So basically, hemoglobin molecule is consist of 4 heme and 1 globin chain. 1 globin is nothing but composed of 4 polypeptide chains. So here it is written 2 pairs of polypeptide is present in globin. So globin means, don't think it is 4 globin. Combination of 4 chain, 2 alpha, 2 beta. As a whole, they are known as a single globin, right? So globin means 4 polypeptide chain, 1 globin. And heme, how many molecules of heme are there? 4 molecules. This is typically depicted here. So this is heme, this is heme, this is heme, this is heme. And combination of all of this polypeptide chain, this is nothing but my globin. 1 globin, 4 heme. Don't confuse, right? So this is the basic structure of hemoglobin. Now, rather than its structure, we have to remember that depending on this globin chain, there are different types of hemoglobin from embryonic stage up to the level of adult stage, right? Before that, you know that alpha globin chain and zeta globin chain, they are in the same family of globin gene. This is known as the alpha globin gene family. Alpha globin gene family, which is coded by chromosome number 16. And there is also another family, which is the beta globin chain family. In this beta globin chain family, what are the chains which is present here? Epsilon, gamma, beta, and delta. So this is the member of this beta globin gene family. And what are the member in the alpha globin gene? Only two members, alpha and zeta. So this beta globin gene chain member are expressed on chromosome number 11, right? So chromosome number 16 contain the genes for alpha globin family, and chromosome number 11 they contain the genes for the beta globin gene family. Now this alpha, okay. So if you look at zeta, zeta can combine with epsilon chain also, zeta can combine with gamma chain also, and both of them will form a embryonic hemoglobin. But if you look at the alpha chain can combine with epsilon also, it can combine with gamma also, it can combine with beta also, it can combine with delta also, and whenever this combination is different types of hemoglobin will forms. Getting my point? So first you look at zeta is interacting with either epsilon or gamma. What is going to form? So this is zeta chain, this is zeta chain. Zeta interacting with epsilon, 2 zeta, 2 epsilon. This is known as Gower 1 hemoglobin. Zeta interacting with 2 gamma chain. This is known as Portland hemoglobin. And alpha interacting with epsilon. Okay, alpha interacting with epsilon. This is known as Gower 2. All of these three varieties of hemoglobin, they are known as embryonic hemoglobin, right? So what is happening? We are talking like this: zeta, alpha, zeta. Okay, it is written in a sequence. And here epsilon, epsilon, gamma. If zeta is combining with epsilon, 2 zeta, 2 epsilon, then this is Gower 1 hemoglobin. 2 alpha, 2 epsilon, then this is Gower 2 hemoglobin. 2 zeta, 2 gamma, this is Portland hemoglobin, right? So we remember like this: zeta, alpha, zeta, Z, A, Z, Z, A, Z, okay, Zaz, Z, A, Z, right? And here we remember E, E, G. So if you arrange them, then you are going to find out this Gower 1, Gower 2 and Portland hemoglobin, right? This is all embryonic hemoglobin. Now fetal hemoglobin, you all know that when 2 alpha combines with 2 gamma, that is the fetal hemoglobin. And fetal hemoglobin start appearing at 8 weeks of gestation, and this is the predominant hemoglobin until birth, right? Until birth, it is the predominant hemoglobin. In case of adult hemoglobin, 2 alpha chain combine with 2 beta chain. We know that, right? Adult hemoglobin major switchover means switchover from this fetal hemoglobin to adult hemoglobin. Previously, we talked about the switchover of hematopoiesis from bones to your liver to bone marrow. But here we are talking about switchover of hemoglobin from fetal hemoglobin to adult hemoglobin. What is the age? 3 weeks of gestation. This is important MCQ. You have to remember, right? Major switchover of hemoglobin from from fetal hemoglobin to adult hemoglobin is occurring 30 weeks of gestation. Then you also remember that at the point of birth, only 20% adult hemoglobin is present and remaining is fetal hemoglobin at the point of birth. But after birth, suddenly this fetal hemoglobin will decrease and adult hemoglobin percentage will increase. And what is the rate? The rate is written here that after birth, fetal hemoglobin decreases 10% decrease for every 2 weeks. And fully adult percentage hemoglobin will reach at 30 weeks following birth. So after birth, whenever the baby is reaching 30 weeks, now mainly adult hemoglobin is present in the blood. And how much percentage? 96%. In these cases, fetal hemoglobin is only less than 2%. So this is all about adult hemoglobin. Now, apart from that, you also know that one minor adult hemoglobin is also present normally in our blood, and that is known as hemoglobin A2. Okay, adult hemoglobin is written as hemoglobin A. And whenever I am saying A2, that is a minor form of adult hemoglobin. Here, 2 alpha chain combines with 2 delta chain, minor chain, right? And it is forming minor adult hemoglobin. Production begins in 35 weeks of gestation, and in adult, the percentage is less than 3.5% in adult blood. If this fraction of hemoglobin A2 is very high in blood, then it indicates the person may be suffering from beta thalassemia, right? So this information which is given in this table, it is depicted in this diagram again very beautifully. Look at in the upper part of this diagram, it is the site of hematopoiesis, right? So it is the site of hematopoiesis here. Okay, already we have seen hematopoiesis. First it starts from yolk sac, then liver and spleen, and then the bone marrow will take over. During this process of hematopoiesis, how the hemoglobin chains are changing. Okay, so first you look at here. What you are finding out, this is zeta, this is epsilon. So when zeta chain combines with epsilon chain, this is forming the major embryonic hemoglobin. But this embryonic hemoglobin, initially it is very high, then this embryonic hemoglobin is disappearing in and around 7 to 8 weeks of gestation. Embryonic hemoglobin disappearing. When this embryonic hemoglobin is disappearing, you can find out two chains are rising its concentration. One is alpha chain, concentration is rising, and this alpha chain concentration, when it is raised to an adult value, it remain in the same way throughout the life. It is remaining same throughout the life, alpha chain. So this is alpha chain. Proportionally, when this embryonic hemoglobin is decreasing, another chain concentration is also rising here. So this chain is also rising its concentration, and this is nothing but your gamma chain. So when alpha is very high, gamma is also very high, they will combine with one another and it will form the fetal hemoglobin. But we know when the baby is reaching towards the point of birth, the fetal hemoglobin concentration will decrease. Why? Because the gamma chain concentration will start decreasing. Whenever this gamma chain concentration is decreasing, I can find out another mirror image chain here. In this diagram, you look at, so this chain, it is like that of the mirror image of this gamma chain, mirror image of this gamma chain. When the gamma chain is falling, the concentration of this green color chain is rising, and this is nothing but your beta chain. So after birth, this beta chain is reaching to a very, very high level, and then it is reaching the adult value and remaining same throughout the life. When beta chain is combining with alpha chain, then it is forming the adult hemoglobin, which is written here, hemoglobin A1 or hemoglobin A. Now there is another chain you have to note down from this diagram. So this is the delta chain. Where the delta chain synthesis started, it has started synthesis from here in and around 35 weeks of gestation. Although it starts synthesized from 35 weeks of gestation, but its concentration is never rising to a very high level. It is remaining very, 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 very low level. When this low level delta chain is combining with alpha chain, which is very high, but delta chain is low, then this is known as HbA2. So this way, you have to remember this diagram, that how these hemoglobin chains are changing their concentration with different stages of life, right? So this is all about the an RBC. Now coming to the WBC. So again the same thing. For WBC, what you have to read mainly the development of the WBC cells, right? So again the same thing, the hematopoietic stem cell is the main one. From there, common myeloid progenitor cell, okay, will be there, and from there the granulocyte GM series cell will be there, and from there the first cell series in the granulopoiesis is nothing but the myeloblast cell. Followed by myeloblast, the next cell is the promyelocyte, right? So all of the feature of myeloblast are written here. Okay, then after promyelocyte, myelocyte, followed by metamyelocyte, then band form, and then segmented neutrophils. So it has shown the neutrophil development. Same way, the other WBC series will also develop. But what I have to remember here, we have seen earlier that more and more maturation of the cell is happening. What will happen? The size of the cell will be reduced, and the nucleus size will also decrease. Hemoglobin is not here, so we don't have to comment about hemoglobin. But look at here. If you look at the size of the cell, myeloblast has a size of 10 to 18 micrometer, but this promyelocyte has a size of 12 to 20 micrometer. Means if I ask you, during WBC development, the largest size cell is which one? Then this is the second one, not the first one. This is exception to the rule. It is not the first cell which is the largest and then size is decreasing gradually. It is the promyelocyte which is the largest cell series in this WBC development. Then the first three cells, that is the myeloblast, promyelocyte and metamyelocyte, they are mitotically active. They can give rise to, they can divide. So mitosis is positive here, here, here. But mitosis is not seen in this metamyelocyte or band form or in case of mature neutrophil, right? Apart from that, you have to remember another important character, that is the presence of granules. So if you think of granulocyte development, okay, or development of WBC, you note down where the primary granules appear and when the secondary granules appear. So look at here. So it is the promyelocyte cells where primary non-specific granule appear. This is very, very important. Okay. Previously, although it is written granules are located, but this is very, very few. First time, specific primary, non-specific primary granule appear in the stage of promyelocyte. Okay, primary granule, non-specific granule. The content of this granule main is MPO, myeloperoxidase. So this is a peroxidase positive granule, right? Peroxidase positive granule. This peroxidase positivity indicate that the cell is developing in the WBC series. That's why this is the marker of this WBC series development cell. Myeloperoxidase positive cell. Okay, that is first time the promyelocyte. Then when it is converting into myelocyte, when it is converting into next cell, myelocyte, now this primary granule is converted into secondary specific granule. Secondary specific granule is MPO negative. So the primary granule is MPO positive, but secondary granule is MPO. Myeloperoxidase is not there, MPO negative. Rather than MPO, it will contain other molecule, like for example, lactoferrin is one of the composition of this granule, and it gives a little pinkish appearance to the granules. But myeloperoxidase granules, this myeloperoxidase is very toxic substances. Along with myeloperoxidase, there are also cathepsins are also there. You may not remember them, but cathepsins are also there inside this primary granule. But secondary granule means myeloperoxidase is negative, but substances like lactoferrin is present in this granule. So these two are also important to remember in this picture, right? Now after that, we all know that in the peripheral blood, various types of neutrophil are there. Don't waste your time here because all of you know this, that neutrophil percentage is 6 to 7%, eosinophil is 1 to 4%, and I hope you know the description of eosinophil and neutrophil. Basophil percentage is very, very less, 0 to 1% only, and the small and large lymphocyte, they constitute 20 to 30% of the total lymphocyte, and monocyte constitute 2 to 8% of total lymphocyte, right? So this lymphocyte and monocyte, they are the A granulocyte, and we all know that these three are the granulocyte. They contain granules in their cytoplasm in mature stage, right? Now after that, if you look at this neutrophil, so neutrophil contain multiple lobes, it contains multiple lobes. More the number of lobes, more mature the neutrophil is. More the lobes, more maturity. There is a counting system by which we count the number of lobes in the neutrophil, and that is known as the RNet count. So RNet count is counting the number of neutrophil with different nuclear lobe and expressing their count as percentage. If only one lobe is present in the neutrophil, that is known as the N1 series. N2, 2 lobes, N3, N4, N5. This way, if you count 100 neutrophil, you express how many cells are there in N1 stage, how many cells are there in N5 stage, and similarly. So maximum cells that you are finding out here is the N3, 3 number of lobes in neutrophil. Now, there is a formula that combined value of N1, N2, N3, if it is more than 80%, then it indicates more immature cells are present in your blood towards the left side, and right side means more towards the maturity. Left side means more towards the immaturity. So suppose there is certain infection in your body. Infection means you have to call all of your lymphocyte for tackling down the infection. So whenever there is bacterial infection, or in case of leukemia, when the cells are not getting places inside the bone marrow, what will happen? More of the neutrophil will enter in the blood. So what will happen? All these neutrophil which are coming in your blood are immature. Immature means less number of lobe. So that's why you are finding out N1, N2, N3 greater than 80%. It indicates left side, and the causes of the left side is bacterial infection as well as leukemia. But whenever you are getting that N4 and N5 cell series are more, greater than 20%, generally it is 10% and it is only 3 to 5%. So 13 to 14%. But if it is greater than 20% combined value, then indicate cells are more mature. More mature cells will be seen whenever the person is suffering from B12 or folic acid deficiency, or the person is suffering from megaloblastic anemia. So this is all about WBC cell series, and the last one is platelet. So platelet, certain important information can be given in your MCQ. Number one, the average size 1 to 5 micrometer. You know that. The volume is 5.8 micrometer cube, and it does not contain any nucleus. Average half-life is 7 to 10 days. All of you know that thing because you have read it in pharmacology. Now the third point is very, very important here. Total platelet, what is present in your blood, 1/3 of the total platelet will remain inside the spleen. That's why whenever you cut the spleen, platelet which was there inside the spleen, that will come to your blood, and what will happen? There will be increase in platelet count in your blood. Okay, platelet count in your blood. That's why it is said that splenectomy leads to thrombocytosis, right? Then platelet, although it does not contain any nucleus, but it contains granules. Okay, one is alpha granules and one is dense granule. What is the content of this granule? That is very, very important for your MCQ. Dense granule contain ADP, ATP, calcium and serotonin. At least you remember this 4 content of the dense granule. Other substances like, for example, your von Willebrand factor, right? Factor 5, factor 8, platelet activating factor, your integrin, cytokine, thrombin, all of these substances, they are present inside the alpha granule. So dense granule contain ADP, ATP, calcium and serotonin. This is important. Major regulator of platelet production, thrombocytosis, is this thrombopoietin, which is synthesized in the liver and other organs. And this synthesis of thrombopoietin increases in presence of inflammation and specific interleukin like interleukin 6. That's why you know that whenever there is infection or inflammation is going on in the body, or interleukin level is 6 is high in the body, then again the platelet count is going to rise or thrombocytosis will happen. So this is all about hematopoiesis and mature blood cells, few information. That is the first chapter of blood physiology.